Good morning, good afternoon, good evening to everyone, whichever part of the world you're in and what's your time zone. Welcome to another webinar, part of the AMM Science and Technology Committee webinar series. And today we'll be discussing characterization of mRNA drug product. Next slide, please. We've been following the same agenda. If you joined us before, we'll have a welcome and logistics, a brief introduction to AMM, and a little bit over the AMM Science and Technology webinars. Then we'll have a background on characterization of mRNA drug products. And we are joined by two speakers, Mandy Johnson from Nano Imaging Services to talk about LNP characterization using Cryo-EM. And we have Wayne Doyle from Eclipse Bio will be talking about sequencing LNP mRNA for characterization. And we'll finish with a roundtable and discussion Q&A. Next slide, please. Please, if you have any questions, post them on the Q&A box during the presentations, and we will discuss and present those questions during the Q&A discussion. Also, the recording will be available after the webinar at the AMM website. Probably give it a week or so before it's posted. Next slide, please. A brief introduction to AMM. Who is AMM? AMM is an abbreviation for Alliance for mRNA Medicine, and it's dedicated to advancing and advocating for mRNA as the next generation modality for therapeutics. The mission is to propel the future of mRNA medicine, and by that, we're looking to help improve patient lives and advocate scientific knowledge to show that and how to harness the mRNA technology for all the advancement in medicine and therapeutics. Next slide, please. So what does AMM do? Like I said, it educate the public and try to remove all these doubts and all the non-true explanation of mRNA for medicine, and also how does mRNA work. It engaged with regulators, policymaker, and stakeholders in North America, actually in worldwide. We have presence in North America, Europe, and Asia Pacific. AMM is developing a comprehensive source of reliable mRNA sector data, and it convenes the community and amplify the work of scientists and organizations through webinars, meetings, and so forth. Next slide, please. The science and technology, AMM Science and Technology, we're dedicated to actually educate people regarding the science. And we have a couple of other webinars coming as part of 9 webinars this year. The next one will be the quality attributes of mRNA drug product and quality control release assays. So this webinar will focus on the characterization. Next webinar will focus on the QC release assays for LNP mRNA. And the last webinar will be December 16 on CMC requirements for mRNA therapeutics. In addition, mark your calendars for April 16 and 17. In collaboration with USP, AMM going to have an in-person meeting on April 14 and 15 of 2026. And this will be a workshop, end-to-end analytical solutions for mRNA therapeutics, supporting manufacturing and CMC need. This will be an in-person and virtual. The abstracts are, request for abstract is open if you're interested in presenting, and also registration will be open very soon. So make sure you keep that on your calendar. Next slide.
To introduce our speakers for today, first we have Khalid Yamut, who is the co-chair of AMM's S&T committee and will be serving as a moderator for today. Our first speaker will be Mandy Jensen. She's the Scientific Director at Nanoimaging Services. And our second speaker will be Wayne Doyle, who is the Head of Scientific Platforms and Strategy at Eclipse Bio.
Okay, so I'm going to walk through a little bit of a characterization of LNP just in terms of the concept of characterization, why it's important to differentiate between characterization and QC release assay. Next slide. So this is probably all of you know this, and I'm just just going quickly over that just to have this in consideration. You have the IVT reaction, you have your input, and your output is a bunch of stuff, which is a possibility, first of all, your mRNA drug substance, but you also have byproducts, uncapped, degraded RNA, RNA abortive fragments, double-stranded RNA DNA hybrids. Now, you try to purify all of that, but then you encapsulate that into an LNP, and that or any delivery vehicle that will go into the nucleus to deliver the payload. So that could be a self-amplified mRNA, your linear mRNA, or a circular mRNA, plus whatever you did not get rid of from your output of the IVT reaction. And it is really important to understand what went into the into your delivery vehicle, and when it goes into the cell, how does these things behave and they impact. Next slide, please. So to add to it is the complexity of LNPs. Here, I put something really quick that was just published recently. This is all the variation of ionizable lipids, helpers, pegylated lipids, and so forth that makes that LNP particle for delivery or a delivery vehicle. And that goes into this, the nucleus and so forth, to get your protein expression or protein translation. Now, each one have a different impact on your product, and each need to be taken into consideration. Does certain functional group on lipids form adducts or they don't, and all of that, and how you control that. So that's an other layer of complexity in terms of the purity of those raw material and how do they interact with the mRNA. Next slide, please. So and to add more to it, here's a representation of, now we're talking about target delivery, and antibodies and proteins and peptides are being, and oligos in certain cases are added to the surface of the delivery vehicles to target certain proteins. So you also have to characterize what did you put on that surface, how many antibodies there is on the particle, is it in the right orientation for binding, and so forth. So next. So this is added another complexity is organ targeting delivery using decorated LNP particles. So that also play into the characterization. Once you characterize all of that, then you can pick what's the best assay to move it to QC release. So with that, next slide, please. Also, you have to consider the stability now of the LNP particle. So there are multiple things that need to be in consideration. For example, your self-amplified, your circular, and the mRNA modification and flexibility of UTRs kind of give support to stabilize mRNA. And once it goes in the LNP, what other things could trigger the stability and the decomposition, not only about the mRNA, but also the LNP particles. So you also have to take that into account. Next slide, please. So here's, in terms of a general thought process would be to develop analytical assays for characterization that could then move to a QC method. So that way, when you have your QC method in QC and you encounter a problem, you can always piggyback to go back onto the characterization data to tell you what that data is telling you if there's something is not right. So method development for characterization of specific attribute. For that, utilize the method for characterization. For that specific attribute, you collect data from various preparation and iteration of the process, how the data correlate with functional assay data, which is really important because you really need to find a link of how the functional assay correlate to the physical characterization. And then correlation with any tox and clinical data from the material that you've tested. And once you have all of that, that could be used to transfer the method to a QC release assay based on all of that data. And this hence the differentiation between characterization and QC release assays. Next slide, please. So for mRNA LNP characterization, I'm listing few here that really have a huge impact, which is particle size and morphology, particle loading, capsule component and impact on delivery, secondary and tertiary structures of the mRNA inside the delivery system, rate of translation, how does the delivery system impact the rate of translation, the rate of degradation, and purity and impurity profiling. The one thing that I did not put here, which is not talked about a lot, and it needs some attention, is the number of particles per ml, because that have an impact on delivery also, and how much you are delivering into the body in terms of the number of particles and how much mRNA per particle, which give you also the dose. Right now, a lot of people, what they do is they remove the capsule and measure the total mRNA in solution. Next slide, please. So functional characterization is really important, and it could be done cell-free assay, in vitro cell-based assay, or in vivo assay, and each have its positives and negatives. So for example, cell-free, it's great to show that you got the target protein, but it really does not account for any cell functions. The in vitro cell-based assay, its translation account for cell function, but limited to the type of cell and not related to organs. In vivo, you get all of the above, but not limited to cell type, and it shows target protein and rate translation in living systems. Next slide, please. So understanding functional characterization. This is something that really how this complicate things quite a bit. We say mRNA is a synthetic molecule. So if you follow the synthetic molecule models, potency can be determined by chemical purity and concentration. That's it. Now you go to biologic large molecules, that's focused relatively well understood and standardized assay. Often is in vitro, in vivo models, simple binding neutralization test. You have the cell proliferation assay and antibody-dependent cell mediation cytotoxicity, and you get your potency there or your functional characterization. Now, mRNA with LNP is a bridge or a combination of both synthetic that lead to a biological molecule. So this is a novel modality presenting unique challenges such as complex intercellular mechanism of action, high sensitivity to degradation, and multiple step pathways for delivery to protein expression. So I view it as it have its own lane that we need to keep in mind for the functional assay for LNP mRNA particles. Next slide, please. So here's a quick case study. This is a case study of cell-free versus cell-based assay. This was done in collaboration with SoCal Biosciences. And what you see in here is 3 different mRNAs were made with one, it's a luciferase. It have 100 A tail, 30, and 0. And what you see on the graphs, the blue is with the cell-free and the green with the cell-based assay. So as you can see with the cell-free, you're not getting this the cellular mechanism and the translation, whether it's GFP or luciferase. It didn't matter what the tail is because the cell action mechanism is not view, it doesn't account for that, the cell-free assay. So your translation is about the same, but you see a difference in translation, between the 100 and the 30, and there is no tail where there's no translation at all. And if you do a Western blot, you exactly see the same thing. So for the luciferase, for example, the first three are the cell-free, which you see a nice band, and then one band for the 100 for the cell base, and you can see it's kind of faded away. Next slide, please. So now it's, if you take that and said, okay, I have my LNP and so forth. So this is the assessing the stability of mRNA LNP products. So those are transfected luciferase RNA LNP in human cells. Samples were collected at 24 hours post-transfection. Luciferase assay activity performed and percent potency compared to the luciferase LNP reference standard, purity of mRNA and LNP confirmed using IPRP HPLC. Next slide, please. So this is, again, work that's been done in collaboration with SoCal Biosciences. The graph to the right is just showed the potency assay at 0, 2 hours, and 4 hours at 50 degrees. And you can see that the relative potency as compared to the reference, which is the LNP particle with mRNA without any exposure to heat or anything like that. So this is what the starting material is. And you have a relative potency of, you don't lose that much at time 0, but at 2 hours, it dropped to 67, and at 4 hours to 64. The graph on the right, it shows you the correlation between the percent purity by IPRP HPLC and the relative potency. And from there, now you can, you do have a system for characterization that if you do something to the particle, how does that translate in the cell, and what does that translate also to the purity, or the purity and the impurity profile of the mRNA inside the LNP particle. Next slide, please. So with that, I'm not going to go through this. This will be available for everyone to look at. I've been compiling this quite a bit, and there's more. I keep trying to update this as a new technology comes out. For characterization, you really need to think about orthogonal methods, because it gives you a huge advantage to understand that if I get the result with method A, I'm getting the same with method B, or method B going to tell me something different than method A, and both of them will give me a nice characterization package and decide which one of those can go to a QC release. Next slide. So in conclusion, one need to differentiate between characterization and QC release testing. Characterization should lay the groundwork for a QC release testing. You need to understand the detailed physical characterization, including the use of orthogonal methods, appropriate functional assays to understand the actual performance, and the correlation between physical characterization and functional assays. And the functional assay could be cell-free, cell-based, in vitro or in vivo assay. And from there, you lay out a roadmap for adaptation of desired characterization assay that will be a QC release assay. And with that, thank you so much, and I'll have Mandy Jansen. It's yours now.
All right, thank you, Colette. That was a really nice laid out overview of LNP characterization. Today here, I would like to talk about a technique that is maybe more niche for a lot of people, but it's being used more and more to assess the LNP morphology, its size, and payload distribution. And I will show here with the help of advanced image analysis tools, these critical quality attributes can also be characterized quantitatively. Next slide, please. So there's two critical quality attributes of LNPs that are size and morphology that are more and more in the spotlight of being having a huge effect on LNP efficacy. So so far, it's been pretty difficult to generate very homogeneous LNP formulations. In fact, all LNP formulations that we at Nanoimaging Services have ever visualized show a certain degree of heterogeneity. So, and we looked over at 1,100 different LNP formulations over the last 4 years alone. The heterogeneity in size and morphology really stems from the lipid composition, the buffer composition. So, for example, the pH, what ionic strengths am I using, the manufacturing methods, so the mixing platform, but also other downstream processing methods. So both size and morphology show an impact on LNP efficacy. And while particle size is routinely measured by tools like DLS, and the total RNA encapsulation is generally analyzed by a ribogreen assay, there's not so much a standard method for morphology that's being used regularly. Since morphology, and in particular phase separation or blebbing, is increasingly shown to be associated with efficacy, direct visualization of particles using Cryo-EM proves not only to be a great orthogonal technique for size and payload distribution, but also to really go into the morphology of these particles. Next slide, please. So what is transmission electron microscopy and how do we use it? So in transmission electron microscopy, electrons are emitted from an electron source under high vacuum and accelerated down the microscope column at accelerating voltages between 80 and 300 kiloelectron volts. And after they pass through the sample, scattered electrons are focused by electromagnetic lenses onto a direct detector device. And because electrons have much shorter wavelengths than light, the obtained resolution using TEM is much greater than with light. So on the right, you can see this diagram, and you can see what additional or how small we can go with TEM. So we can see viruses, we can see LNPs, but in these LNPs, we can also look at the double leaflet of the lipid bilayer. We can look at individual proteins. So it's a really nice like visualization technique to look at these smaller entities. Next, please. Oh, so this is one of the 6 microscopes we have at Nano Imaging Services. It's 11 feet tall, so it's huge, but this is how nowadays these modern microscopes look. Next, please. And these are just typical particles that we look at a daily basis. So you can see a range of different nanoparticles. What they have in common is that they all need to be in an aqueous solution for us to visualize them. And generally, the particles have to be between 5 nanometers and a micron in size. And for people who are familiar with TEM, you know that we can visualize particles either using negative stain or by vitrifying them in a thin layer of ice called Cryo-EM. Next, please. So when we use negative stain, particles are embedded in a heavy metal salt, and this gives really, really nice contrast to especially smaller particles, but the sample is dried and you see a collapse of structure. Also, what you're imaging is an envelope of stain around these particles. In vitrification, you embed particles in vitreous ice. This gives a little poorer contrast because you don't work with any stains or labels, but you preserve these particles in a near native, like hydrated condition. So on the right, you can see 2 examples. The left 2 panels, top and bottom, that's a SARS-CoV-2 protein. You can see a negative stain on the top. These particles are very nicely shown. They have a lot of contrast. But if you look on the right, LNPs in negative stain, these particles, yeah, I see them, but the morphology is not maintained, and I can also not see any of the internal structure. So on the bottom right image, you can see LNPs that are actually preserved in ice. And these particles, you can see that the morphology and the size is well maintained. You can see the internal structure of these particles. You can see phase separation in some of these particles with an aqueous phase and a lipid phase. Sometimes you can see even in this lipid phase some mottled appearance, which is either attributable to the hexagonal packing of lipids or to mRNA present in these particles. Next slide, please. So for Cryo-EM sample preparation, We use a robot, but in principle, you add 3 microliters of sample to a 3 millimeter copper grid, and you blot off most of this sample until a very, very thin layer of sample is left on this grid. And this you plunge freeze very rapidly into liquid ethane that is cooled down to liquid nitrogen temperature. So the freezing of this ice layer goes so fast that the ice forms amorphous ice that you can look through. So it's like almost glass-like, in a glass-like state. So these particles are then preserved, the lipid nanoparticles are preserved very nicely in this lipid ice layer. Next. This is the robot that is generally used. It's a VitroBot. We can precisely tune blotting parameters, also temperature, humidity, etc. So this ensures that we can make very consistent grids with ideal ice thicknesses. Next, please. We also use automated image acquisition packages where we start at very low magnification. We image the grid and we look for optimal ice on the grid. And then we progressively increase the magnification until we reach a magnification where we can actually see the nice features of these particles and can start looking at the critical attributes of these particles. Next, please. So the advantages of Cryo-EM are that we can visualize LNPs at high resolution and at the per particle level. These particles are, as I said before, frozen in their near native hydrated state. We only use 3 microliter of sample to make one grid. Lipid concentrations need to be higher than 10 to the 11th particles per mil. That usually is the same as like 1 to 10 mg per mil lipid concentration for LNPs. The technique is not affected by impurities, and we can get multiple quality attributes from the same data set. So we can look at size and homogeneity of these particles. We can look at shape, at the lamellarity, if there's phase separation, so bleb formation. We can measure bilayer thicknesses in these particles. We can estimate PEG layer thickness. We can look at the payload distribution. Did the RNA, for example, migrate from the lipid phase to the aqueous phase? And obviously, we can see if anything weird or unexpected is going on with these samples. When we combine Cryo-EM imaging with image analysis tools, we can actually get quantitative data out of these data sets. Next, please. So Cryo-EM image analysis generally requires image pre-processing and normalization of the data set, then particle picking and segmentation, using computer visualization techniques such as edge detection. And then generally, we also add filters to remove false positives, such as ice contamination. The picked particles and the traces are then either used for size distribution analysis, where we use the traces to calculate size metrics from the data, or we use the particle extraction coordinates to then feed into other program, which uses models that we generate to do particle classification. Next, please. So this is an example of like the size distribution analysis. So we can trace all these particles in an image automatically, and we use all images in a data set, and we can at the same time like automatically process thousands of particles if needed. And from these traces, we can get size metrics such as area, equivalent diameters, minimum and maximum thread diameters, circularity, but also derivatives as, you know, the d-values, the span, aspect ratio, etc. And if we do that over multiple grids, so on the bottom right, you can see we did it, we made 3 different grids of the same sample, and you can see that the repeatability of these size measurements is pretty good. Next, please. If we look at particle classification analysis, we can use the same data set, the same images, so using the same thousands of particles if needed, to look at each particle and assign a class. So we can classify these particles based on, for example, their morphology. So we can look at how many particles in a sample are, or in the images are unilamellar. How many particles show phase separation, or blebbing? How many particles show mRNA that have moved into the aqueous phase, for example? And in the panel on the right, these 12 smaller images show you different types of LNPs that we regularly see in LNP formulations. And we can categorize based on lamellarity, on blebbing, on shape, as I said, but also on the payload distribution to detect where we see the modeled appearance, whether it is in the lipid phase or in the aqueous phase. Next, please. So combining Cryo-EM with these advanced image analysis tools gives a really nice way of characterizing these LNP formulations, especially when formulating new LNP samples, when going through process optimization, but also when you look at stability under different conditions. So I will show you a couple of examples just to show you how Cryo-EM can be used. And we basically came up with these examples together with Phosphorex, and Phosphorex generated these LNPs. The LNP formulation that is used in the following examples are a basic standard LNP formulation of the 4 standard lipids. Next, please. So in the first example, we looked at comparing 4 different mixing platforms. So particles were generated using a T-mixer, an Ignite Plus, a Jet mixer, and a Sunshine. And we took images, like a full data set of each of these samples, and we did particle size analysis on each of these. And we saw that the Ignite, the Jet mixer, and the Sunshine gave very similar results, particle size results. They're all about 60 nanometers. The T-mixer is gave slightly smaller particles. But more interesting for us was that Cryo-EM really showed the differences in morphology here, because while the Ignite, the Jet mixer, and the Sunshine showed 40% to 46% of particles that showed phase separation or bleb features, the T-mixer showed about half of this percentage. So only 27% of the particles generated by the T-mixer showed blebbing. Next, please. So this is the second example where we looked at a formulation. So in the top left corner, you can see an image, a representative image of a formulation that was like LNPs were generated and then dialyzed into a low salt pH 7 buffer. The payload was 6,000 nucleotides. And then we compared that to a couple of different processes or formulation changes. So if we look at the same formulation, but not dialyzed into the low salt pH 7 buffer, but into a high salt buffer, we can see that, we can see that the size of the particles is a little bit smaller than in the low salt buffer. But more importantly, the blebbing goes down from 46% to 1%. So this is a huge difference that can easily be detected by Cryo-EM. If we not use Cryo-EM, or if we don't use dialysis to get it into the low salt buffer, but TFF, we can see that the particle size is slightly bigger and the blebbing is slightly increased. If we look at an LNP formulation with a slightly smaller payload, 800 nucleotides instead of 6,000 nucleotides, we can see that the overall mean diameter does not change, but the blebbing does change from 46 to 11%. So these are things that can be easily detected using Cryo-EM and image analysis tools. Next, please. Here is just an example of 2 of these conditions where we made 3 different grids instead of just 1 grid. And you can see that in both cases, the size is very repeatable, as well as the measurement of the percentage of the bleb features. Next, please. In this last example, we looked at a LNP formulation immediately after we received it at NAS. So that's the image on the left. This was imaged within a week after formulation. And then the two images on the right are representative images of LNPs that we imaged 10 weeks after we received it. So we kept it at 4 degrees, and then after 10 weeks, we imaged it again. And the main difference is that you can see much more blebbing. You can see that the blebs are bigger. Some of the particles are bigger. They also show more blebbing. And in general, like the whole particle distribution is wider. Next, please. And just because a lot of people always ask us, like, if the Cryo-EM analysis, the particle size analysis, compares very well with DLS, we asked Phosphorix to also measure the size of all these formulations that they send us with DLS, and we plotted that against the size that we got from the Cryo-EM data. And you can see that the Cryo-EM data and the DLS data, they correlate very well, but the size that is measured with Cryo-EM is overall higher than what we measure with DLS, which is not very surprising because of the hydrodynamic diameter of these particles. And also, you know, bigger particles scatter more than smaller particles, so they add much more to the signal in general. So this is not surprising, but I just wanted to point it out that generally the correlation it's very nice. Next, please. So as I have shown you, you can get a lot of information out of 2D images if you combine it with quantitative analysis or image analysis tools. You can get even numbers out of these to quantitatively say something about the quality attributes. But sometimes, especially when you first start looking at your LNP formulation, you may have additional questions that can be very well solved looking at the 3D volume of your sample. So on the left side, you can see an image which is just a 2D image, which is in this case doxorubicin within liposomal particles. And you can see that the liposomal, the doxorubicin crystals are these very lengthy crystals within the LNPs, but you can also see these very dense, round interior densities within particles that at first sight you may not know what this is. So if you apply something like cryo-electron tomography, where you add or you put the sample in the microscope and you turn this sample, you rotate the sample from, say, -50 to +50 degrees, you take an image every 2 degrees, do a back projection, and you can now get a 3D volume of your sample. Can you play the movie, please, Noor? Thank you. You can now see that, for example, this very dense density within the particle is actually a top-down view of a doxorubicin crystal within the particle. You can also see with the green arrow, if you play it again, that a multilamellar particle is really a multilamellar particle and not two particles on top of each other. So these are kind of the questions that you can get out of a 3D volume. Next, please. So in summary, Cryo-EM is a direct visualization per particle analysis technique that requires only very little sample and is compatible with a wide variety of buffers. It provides information on a variety of quality attributes such as size, morphology, and payload distribution. If you combine it with image analysis tools, you can get quantitative data that you can use for statistical evaluation of your samples and compare samples to each other. And we often see LNPs come in and clients use it for characterization during drug formulation, process optimization, as well as assess stability and consistency of their samples. Next, please. And this is my last slide. At NIS, we have 6 microscopes at 2 different sites. We are GMP compliant in our San Diego site, And I'm very happy to answer any questions at the end of the presentation. Thank you very much for listening.
So I'll get started on my part of the presentation. I want to first thank AMM for the opportunity to present today. And so over the past few years, we've seen incredible advances in mRNA medicines, and LNPs have been at the center of that success. But as we move to the next generation of therapies, one of the biggest challenges that has emerged is characterizing what's actually happening to the RNA inside of these lipid nanoparticles. And so sequencing-based approaches can help provide details on this. And I'm going to first just say that I work at Eclipse Bio, and what we do is we develop different types of sequencing assays and analytics that help advance RNA therapeutics. And we apply this sequencing-based support across the entire RNA drug development process, from design to optimization. And if we go to the next slide, I'll just mention that the sequencing-based approaches I'm going to discuss today have been applied to support things like protein replacement therapies and other therapies. But first, looking at sequencing itself, I just want to kind of give a brief overview of what it is. So sequencing starts by isolating the RNA. That can be an RNA that's already probed, has some type of enrichment on it, such as enriching for poly-A tails. And then if we look at a traditional Illumina-based sequencing approach, the RNA gets fragmented, and then you prepare sequencing libraries. This is done by converting it to a cDNA, and then putting on a sequencing machine that gives a nucleotide level readout of what bases are present. And then we can do this for typical kind of RNA-seq approach, where you look at the level of an RNA that's present. But depending on how you prepare the RNA, you can also look at things such as secondary structure, what modifications are present, regulatory factor bindings such as microRNAs, or the actual dynamics of translation. And so these types of measurements then allow us to go far beyond some of the traditional assays and really understand how the RNA is behaving on a molecular level, including within an actual drug product itself. And so if we go to the next slide, as we discussed throughout this webinar, there's multiple aspects that need to be characterized to look at how an LNP mRNA is behaving, from understanding are the correct lipids present, as we just saw, understanding the morphology of the LNPs, as well as are they encapsulated and how is the RNA behaving itself. And there are a number of very powerful approaches for this, including, if we go to the next slide, many of these can also be characterized using sequencing as well. And so for today, I'm just going to show two short vignettes about how sequencing can be used to look at, is the RNA actually remaining intact within the LNP, and then what's the rate of the actual uptake. So starting first with uptake of the LNP, intactness of the RNA, which if we move to the next slide. One of the biggest factors that will drive mRNA stability, both in solution and inside the LNP, is the actual structure of the RNA. And because molecules are very dynamic, they can fold into highly structured regions where bases are paired, as we can see at the next animation. And this increases the stability. Things like in helices, this is going to make it less prone to hydrolysis, and the RNA can remain intact and productive longer. Or with the next animation, if you have unpaired bases, this can decrease stability. These are all sites that can be prone to hydrolysis and lead to a less functional protein product. And so on the right animation, you can then see from a linear design paper that the more structured you have an RNA, so the more base pairing that's occurring, the longer the half-life of the RNA will be. So we're going from 20 hours down to, if you decrease the structure, have less base pairing, you can get a half-life of down to 3.9 hours. And so while structure is very important, if we go to the next slide, we can see kind of the typical approach that's used to determine structure as part of the design process, in which you'll take your RNA sequence of interest that encodes your protein product, and then use a computational algorithm to predict what its structure will be. This is based typically off minimizing the free energy with thermodynamic principles, so trying to maximize the amount of base pairing. And then you get a nice looking structure like shown here on the right, in that there's base pairing, there's unpaired loops present as well. And then you have a report on what's called the minimum free energy, where the more negative it is, that indicates that it likely will be more stable. But importantly, this is a prediction, and this is based off how an RNA would fold just based off its sequence and kind of neutral buffer conditions. But if we go to the next slide, within an LNP, this isn't the case, because the RNA structure is going to be affected by excipient interactions, including what lipids are present, buffers that are present as well. And an algorithm then can't account for all these interactions that are occurring within the complex environment of the LNP. And so the RNA structure that you think you have based off that prediction that should be stable can be completely different with the LNP, making it more prone to hydrolysis and less effective once it actually enters the cell. And so going on to the next slide. So since we do know that the encapsulated RNAs can fold into unique structures, it's then important to actually measure what's occurring. But some of the approaches, as shown on the animation on the right, do have limitations when actually used with an LNP. For example, spectroscopy can be used to look at the structure of an RNA. It's very effective in free, unencapsulated RNA, but then you get interference from cholesterol within LNPs if you attempt to use this actually within the lipid nanoparticle itself. And so moving on to the next slide, what we've approached is using sequencing to overcome these limitations, in which we directly measure what bases are accessible and unpaired versus paired while the RNA remains encapsulated directly within the LNP. So we first maintain the intact encapsulated RNA. We don't remove the RNA out. We don't denature it. And then we treat with a chemical reagent that interacts with any base that's unpaired. So anything that's within a loop is shown here, it's able to interact with. It then adds a small chemical addict to it. And then when we do next generation sequencing, shown here on the right, when we convert to a cDNA library, that chemical addict leads to a mutation. And we're showing that here, where along the x-axis is each nucleotide position. And the amount of color indicates the amount that we're determining mutation is present. So in other words, the amount that that base is actually accessible within the drug product itself. And so in the middle, we have a lot of increasing color. That's a region that's unpaired, a large loop that's present. Where we have that drop in color, for example, more to the left, that's where a helix is present and it's more stable RNA that's present. And so we can then use this to look within LNPs. I'm just going to show an example here on the next slide, in which we did work in partnership with Acutus Therapeutics, and that they had two LNP formulations. LNP formulation A was leading to a very stable RNA. It could remain in storage for a long period of time. It remained very potent. They weren't seeing any fragmentation patterns when using their measurements. Well, LNP formulation B did lead to degradation very rapidly in storage. And so they got fragmented RNAs and less protein production at the end. And then what was unclear, if we move to the next animation, is what was actually the mechanism behind this degradation. And so the hypothesis was this was due to a structural change due to the excipients that were present, making specific regions more prone to undergoing hydrolysis. And so we move on to the next slide. We approach this by using sequencing to look at how the RNA is actually behaving. So on the left, I'm showing an unencapsulated RNA. So this is how the RNA folds naturally within buffer conditions. And this is again using that chemical probing. So directly looking at the structure, this is one small subregion. In red are unpaired bases. And so these are sites that could be prone to hydrolysis. And then in the dark teal are paired bases. So these are going to be less prone to actual breakage. And overall, we're seeing a mixture of unpaired and paired bases. This is normal for an RNA. There's naturally going to be some unpaired regions. You want to have that also for protein production, but overall, a fairly stable structure. If we move on to the next animation, this is LNP formulation A, which is the stable drug product. And it has essentially the same exact structure that we measured, again, looking directly within the LNP itself. There's a slight switch that a loop has dropped down, but otherwise this is essentially forming the exact same amount of base pairing with unpaired and paired bases. If we then move on to the next animation to the unstable product, this is LNP formulation B that was leading to rapid degradation. And we found that the majority of the bases in this region are then unpaired. And so you can see that here, where they're very reactive, the chemical is able to interact with them. And the only real structure that can form are these two large loops. And each of these red positions is then a point that's going to be more likely to undergo hydrolysis as it's in storage over time, leading to less efficacy once it actually enters the target tissue. And so if we move on to the next slide, a benefit of this then is that we can look directly within the LNP, see what the RNA structure is, which means that we can save a lot of time. We don't need to do as long of weeks to months of testing to see is fragmentation actually occurring. Immediately after encapsulation, sequencing can be used to see are there unstable structures forming, and does that change between different types of formulations? And then importantly, I'm not going to go through it today, but there can also be orthogonal approaches, including sequencing-based, to say, do we actually see fragmentation occurring where we see these structural changes, supporting that these are sites that are going to become problematic when it's used in actual production. And so that was one example of how sequencing can be used, focused on the structural component. On the next slide, I'm now going to move into, once we've characterized the RNA within the LNP, how does the drug product actually behave in being taken up by the target cell type? Is it able to then escape into the cytoplasm associated with ribosomes? And how long does it remain actually functional within the cell type of interest? And so this is related to some of the limitations of current potency assays, in which we'll use something to look like a Western blot or an ELISA and look to see, is the protein of interest actually being produced? And while these are very effective, it can have some limitations. There can be issues that you don't see high protein production, and that's unclear. Is that due to limited uptake into the target cell type? We often can see some cells, they just don't really express a protein once it's encapsulated within an LNP. This also could be due to poor release, that it's actually taken up, it's within endosomes, but it's not able to escape out. Or as mentioned earlier, there can be adducts that form. This can lead to pauses, preventing actual protein production from occurring. And none of these are really detectable by using a Western blot or an ELISA-based approach. So what we do is shown on the next slide, in that you can use an optimized ribosome profiling to actually look at the dynamics of translation and see what's actually occurring. Is it actually escaping into the cytoplasm? Are there stalls and other features of ribosome dynamics? And so for this ribosome profiling or RiboSeq approach, a sample is transfected with the drug product, and then that sample is split in half. For half of it, we extract out the RNA and do a typical RNA sequencing library. So this gives us the relative amount of RNA that's present within the cell, giving an idea of how much is actually being taken up. And then for the other half, the ribosomes are locked in place, and then all of the free RNA is digested. So the only RNA that's going to be left is directly within the ribosome complex, giving nucleotide level positioning of where translation is occurring, but also allowing us to see, did it actually escape from the endosomes, and is it now able to associate with ribosomes and produce protein product? Sequencing can then be performed in a variety of different analyses shown on the next animation, including, for example, here, where we're looking along the length of an RNA, and then the height is showing the amount of ribosomes at each position, allowing us to actually visualize the dynamics of ribosome translation along the length of the RNA, or identify stalls that could be induced by lipid adducts. On the next slide, I'm just going to show one example type of analysis that's being done with this type of RiboSeq approach, which is to look at the dynamics of translation over time. And so the experiment that I'm going to show is where an RNA was transfected into a cell line. In this case, it's going to be HEC293 cells. And then the amount of RNA within the cells and the amount of translation over time is examined. I'm looking at time points of 1 hour, 2, 4, 8, and 24. Importantly, we're going to do a washout at 4 hours. And so this allows us up to the 4-hour time point to look at the amount of uptake that's occurring. And then after 4 hours, that allows us to examine kind of the overall stability and the overall amount of time that the protein can actually be produced. And so if we go on to the next slide, first looking at the RNA sequencing data. So this is telling us the amount of RNA that's actually taken up and that remains within the cell type of interest. We're showing a time course of that over here on the right, where the y-axis is the normalized amount of RNA that's present in relation to all of the expressed genes within the cell. And so even with an hour, we start to see uptake occurring. That continues over the course of the first 4 hours to when we do the media washout. This remains a very steep curve, so expect if we were to go out further, the RNA would continue to be taken up. After the washout, the RNA then remains in the cell stably for another 2 hours, at which point it starts to degrade as the tail begins to be lost, and then RNases start to chew up the RNA, limiting the potential amount of protein that could be produced. So this overall gives us an idea this is an effective way that it can be taken up into a cell, and then the overall time course that it's going to be available. We also want to confirm, is it actually escaping into the cytoplasm, actually associating with ribosomes. And for that, we can go to the next slide, which looks at the ribosome profiling data, which again is looking at the RNA that's actually associated with ribosomes. We can see at all time points, the purple curve is higher. This indicates that there are multiple ribosomes for every copy of an RNA, so it's being very efficiently translated. And within an hour of it being introduced, RNA has escaped into the cytoplasm. It's able to associate with ribosomes. And then even once the media is washed out, the ribosomes continue to associate with the RNA, continue to translate it. It remains stable for a longer period of time than the RNA itself, because it's able to still be very efficiently translating. Even as some populations of RNAs become degraded, the cell increases its overall amount of translation to continue to produce. Although by the 24-hour time point, it begins to decay down as well, as the amount of functional RNA decreases and there's less available to actually produce the protein product. And so this then allows us to look at both what is the actual uptake, what is the actual time to endosomal escape, and then what is the time that actually remains present within the cell to produce a protein product and be functionally relevant. And so if we go on to the next slide, this type of sequencing-based approach then enables us to do analyses similar to here, where we can look at the uptake and release of a single drug product. But these sequencing-based approaches can also be used to examine changes in the actual drug product handling, if it's shaken, if it's lyophilized, if it undergoes freeze-thaw, or is at different doses. How does that change the actual uptake, the actual release? And then something that's also becoming more of interest as we move into kind of more next generation therapies is looking at how do these dynamics change if you have more than one RNA payload within the same LNP. There have been effects reported that you prepare two separate LNPs, each with a single RNA. You'll see very efficient translation of both, even if co-delivered. But when both RNAs are introduced into the same LNP, you can start to see effects that only one RNA is well translated while the other isn't. And this type of sequencing-based approach can then tease apart what's actually occurring there to lead to those changes in translation. And so moving on to the next slide. What I showed today was just two examples of looking at sequencing, looking at how does the RNA remain intact within the LNP and how is its uptake and escape. But sequencing can also be used to look at other dimensions, such as is the cell responding with TL4 responses or other unexpected effects for the specific drug product, as well as the actual identity of the RNA. Is it correct within the LNP itself? And then moving on to the next slide, that's also just one small slice that sequencing can actually provide for characterizing both the drug substance and the drug product. Sequencing really can be used to look at a majority of critical quality attributes, from the actual identity, both in terms of sequence and structure, to poly-A tail length, the actual stability from drug substance to drug product, like shown today, to within the cell, and integrity assessments as well. And so for my final slide, I just want to mention that everything I shared today fits into a bigger theme of how sequencing really can accelerate RNA drug development. And at Eclipse Bio, we had built a platform that's sequencing-based to go from AI-powered design through drug product characterization. And so whether you're designing next-generation mRNA vaccine or optimizing a therapeutic RNA, such as a gene therapy, sequencing can give insights to allow you to move faster and with more confidence to support some of those release assays that are then used later. And so kind of just to go to the next slide for an overall conclusion, I'd really like to thank everybody for the time for listening today. And if you'd like to learn more about how sequencing can support your characterization needs, you can also reach out to Eclipse Bio at info@eclipsebio.com.
Okay, I think you have to unmute yourself.
Okay. Okay. Thank you both for a very, very informative presentations, and lot of these things are not what people are looking at these days. They're going with the generic or the much well-known physical characterizations, all this. It's really interesting and exciting. There's a couple of questions I want to go through right now. So for example, one question is for Mandy. Cryo-EM is a powerful tool to QC LNP mRNA, but the equipment is expensive and not very manufacturing plants have access. So what are the benefits of using Cryo-EM, and what are other industries recognized methods?
Yeah, it totally, that's a good question. And I realize, Cryo-EM is not a high throughput method, and it is fairly expensive, but it's the only direct visualization method of, it's a per particle technique. It's very different from other bulk technique, and it's in that regard a very good orthogonal method, because you see things, you directly visualize things that you wouldn't otherwise see. There are other ways to visualize, obviously, but you need fluorescent tags for that, and it's not always high res. So in this case, you can immediately see the internal structures of these particles, anything that may be wrong, distribution of payloads. It's in short a very highly effective orthogonal tool to visualize not bulk samples not in bulk, basically.
I would like to add to that and this is the whole reason when I thought of doing these webinars to separate characterization from QC release testing, I view Cryo-EM as a more of a characterization tool that is used to finalize your formulation, understanding how your LNP is reinformed and in what kind of morphology, and how much with a certain technique, how much you have, how much mRNA there is encapsulated, is anything is out there, and so forth. And this is really important to understand, that there is a difference between characterization and QC release, because once you have this characterization, you can go to an orthogonal method, kind of like what you showed, Mandy, in terms of the correlation with DSL. So once you have that formulation, you understand the morphology, you got the DSL data. So the DSL is more QC friendly and affordable, but you form that bridge. So in case you see something, you can go back to cryo to understand if something have changed. Thanks for that. I do have a question from my end. Just want to follow up on that. You showed some interesting results regarding the particle size. It changes between dialysis and TFF buffer exchange. Is that due to the Oswald ripening, or what is happening? Because this is more of a now particle chemistry issue and not an mRNA issue, because we're using particles to deliver. Do you think that's a Oswald ripening, that there's such small particles in there that are being disruptive and added to the more stable particles to give you a larger size?
Yeah, that could very well be, but Cryo-EM is a snapshot, so
Okay.
it's very hard to follow what's going on. So it's a snapshot in time. So I can't say too much about what is going on, if it's fusion events or yeah.
Okay.
Yeah.
Wayne, this is going toward you. Is so understand compact RNA is more stable. Does a more compact mRNA makes ribosomes less accessible?
Yes, so that is a major challenge, is that they're kind of two sides of the same coin, that the more stable kind of structure you have, the more stability you have, but then that leads to overall less translation in terms of kind of a burst of protein production. So typically there need to be strike a balance between enabling ribosomes to come in and enter, but enough structure that remains stable. That being said, that really depends on the goals. There's some approaches that you just want a sudden burst of protein production as rapidly as possible. If it's less stable, that's actually a good thing. While others want it to be as stable for as long as possible and kind of a slow trickle of protein production. is okay. So it really depends on kind of the goals overall between it.
Yeah. And as a follow-up to that is, does your sequencing that you provide, does it distinguish between uridine versus pseudouridine or N-methyl pseudouridine?
Yeah, so that depends really on the type of sequencing approach that's being used. So for structure profiling, for example, some of the translation that I showed, that really can be done on uridine, pseudouridine. There's some changes in reactivity that can occur. But if you want to identify specifically which bases are modified or unmodified, there can be other sequencing-based approaches. For example, nanopore sequencing looks at the direct RNA and then uses an electrical current to see what bases are present. So that can be used to identify, is this a uridine? Is this a pseudouridine alternatively?
Yeah. Back to Cryo-EM. Let's see. Oh, just one. So when the formulation does not contain 1 mg per ml lipids, is enrichment needed, and how is this done, and are there any consequences for the LNP morphology?
Yeah, that's another good question. So if the formulation is really low in particle concentration, there's only so much we can do with Cryo-EM. So we glow discharge grids to make them charged. So we can play with that to get more particles on the grid, or we can apply sample multiple times to the grid to try and enrich particles on the grid themselves. But that would not affect the morphology of the particles. I think going the other way around, that is more where the particle concentration is too high for Cryo-EM. In that case, we would have to dilute out the particles, which also would not do anything to the morphology. But if there's, for example, too much sucrose in the sample and we have to dilute the sucrose out, then that would obviously have to be done by either dialysis or a quick dilution, and the dialysis in itself may affect the morphology. But in general, like enriching particles on the grid or diluting particles like just before grid preparation, we've never seen that affecting the morphology or the size of the particles.
So you're going to jump back to sequencing. When base variations are observed through sequencing, can you tell where the variation comes from? And is it because of the mRNA molecular, or is it due to reverse transcriptase or NGS sequencing steps? And I would like to add to that is, can you determine fidelity out of those sequences?
Yeah, so for the structure profiling that I showed today, we always include a control as well that has not been treated with anything, and so that can give us kind of an understanding on over cross time and multiple samples. We've done this repeatedly, so we've looked at kind of what is the amount that arises from RT, what's the amount that arises from the sequencing, and then we can use that and look at individual samples and say, is that kind of above expectations? And so we've used that to look at fidelity of the plasmid itself. We've been able to see that there's very high rates of the wrong base being present, very consistent with the plasmid is incorrect. But we also have used that at kind of lower levels to say there might be some issues with kind of the actual fidelity of the polymerase itself. That's from the Illumina base, which does go through that cDNA component. This can then be paired with direct RNA sequencing and see as an orthogonal measurement, does that also show kind of incorrect base incorporation of the same sites? We have done that, for example, comparing manufacturers. We're able to see with both cDNA-based sequencing and direct RNA sequencing, kind of the incorrect base being present, really consistent that it's the mRNA itself. Teasing apart, is it the polymerase fidelity versus the plasmid fidelity? Really comes down to looking at the amount of misincorporated base that you see. When it's at a lower level, that's more indicative of T7 having an issue.
Okay. Can you hear me? I think, okay, good, because something happened with my computer and now, okay. For TEM, how many particles would you image to remove any potential bias? Because you know that I've done TM in my past. You know, it's kind of tricky because you're taking a very small sample out of a large amount in order to do that. So
Right. So what we generally do is look at a minimum of 250 or 300 particles to do an analysis. This is, how do you say that? I showed in the slides that this is fairly repeatable, like over, if you look at 3 different grids regarding size, but also morphology. If we were to do this, how do you say that, as as a validated method, we look at many more particles. We look at a couple thousand particles, and the number of particles really depends on the heterogeneity of the sample. So the more heterogeneous your sample is, the higher number of particles are needed. But generally, a couple of thousands for those is enough.
and also with Cryo-EM, would you be able to see any antibodies that is decorated on the particle? You know, or can you determine if there's an antibody conjugated on the LNPs?
Yeah, that depends. So if it's the full antibody, technically it's 150 kD, we should be able to see it, but it also depends on the conjugation efficiency. So if it's just 1 or 2 antibodies on the outside of an LNP, it's very hard to detect based on the orientation of the particles. We have had client samples that had a very high efficiency of antibody labeling, and in those cases you can definitely see it. And we did, for example, cryo-electron tomography to see in 3D, and that's pretty amazing to see.
Yeah. Wayne, regarding your sequencing, what is the behavior that you see between monovalent and bivalent preparation in the LNP particle?
Yeah, so within the LNP particle itself, the approaches we've looked at that has really been in terms of the actual kind of uptake and delivery component. And so we had a partner that was producing monovalent preparations. So they had one RNA in one LNP, another RNA in another LNP, delivered it, and were able to see efficient translation of both. But then as soon as they added both to the same LNP, they saw very inefficient translation of one. That does appear to be some aspects of kind of codon optimization, that one RNA is a little bit more codon optimized. And so that kind of sucks up a lot of the ribosomes and the other one gets a little bit penalized. We do expect, though, that there's some structural components of that some interactions occur within the LNP that continue through as well. And that kind of you have a mixture of both of those effects occurring.
Now this question is for both of you. Most of what got presented today is based on LNP, and I know LNP is, you know, what is used quite a bit right now. But have you applied either of your techniques to a protein, polymer-based particles, or other modality than just lipid nanoparticles?
Sure, I can go first. Yes. Yeah, we look at a lot of different nanoparticles. Basically, anything that is between 5 nanometers and a micron, we image. So there's indeed a lot of polymeric nanoparticles, either with DNA or RNA. Also, a lot of gene delivery vehicles, such as VLPs, AAVs, lentivirus. And in those cases, we look also at empty full aggregation, impurities. So we do, yeah, an inorganic material as well, but it's, yeah, definitely.
Okay. How about you, Wayne?
Yeah,
I mean, would the sequencing get applied also there?
it would. In terms of especially looking at the uptake and the delivery, the only one that I would need to check with our internal team about is looking at structure within those to see if the reagent can actually enter, for example, a more complex nanoparticle. We need to do some testing to optimize and make sure that that works as expected.
Okay. All right. Thank you. So, for the sequencing, is the only effect of LNPs on RNA translation related to uptake and endosomal escape, or are there other ways LNPs can change protein production?
Yeah, so I briefly mentioned it. I'd say the other main one that we've gotten a lot of interest in is adduct formation. So if an adduct is on a base, when the ribosome reaches that position, it's not going to be able to interpret really what should be going on there, and so it can't bring in the correct tRNA, and so that can lead to stalls, preventing the actual protein production from occurring. And so we do have an interest internally on kind of approaching to see where those addicts located, are those relating to the actual stalls that come out at the end of the day.
Okay. Now, Mandy, in terms of LNPs are stored in, sometimes people add additives to prevent them from freezing because it's stored at -80. Uh, so such protecting as, let's say, sucrose, does this affect the imaging, or are there any other buffer components that affect imaging? If so, how do you deal with these?
Yeah, it's usually the sugars, so sucrose, trehalose, that affect the image quality or the contrast within the images. So it becomes more difficult to see the particles. We usually image as is, so 10 to 15% we regularly see come by, and the sizing is fine. But if you look at internal structures, it becomes a little bit more difficult. And in those cases, we would dilute the sample in a buffer without those additives immediately prior to grid preparation, and look at the samples in that way as well.
So in general, this question going to both of you. How likely these two different techniques you think that will be taken on and help push the envelope in terms of characterization of LNP mRNA? Whoever want to go
I can
first, so.
I guess I can start with this one. We are seeing that sequencing is becoming more and more adapted. It really does allow for really insightful characterization on the characterization component, kind of really insightful in multiple dimensions on critical quality attributes in one go. So definitely in the characterization space, we see that continuing to grow. We've heard that from partners at conferences, from regulatory bodies as well. I'd say in the release space, that becomes kind of a little bit more of an open question. Do you want that much detail at that point, or do you use it more to validate and then set established metrics than you use during the release stages? So I'd say that's more, can go in multiple directions in the future.
Yeah, and for Cryo-EM, it's as I said in my first slide, it's more and more being used. We've seen in a very high increase in the last couple of years, obviously, but it's also one of the only techniques where you can look inside of the particles. You can see what is happening with the payload distribution. So we're not getting requests to characterize these LNPs early on, but also later on in stages, especially because we're now doing advanced image analysis and the subjectivity is taken away from analyzing these images. So we'll see. I think Cryo-EM will be extremely important looking at these type of particles.
Back to some questions that have been posted. So this goes to you, Wayne. To different RNA modification in IVT changes the RNA secondary structure and ribosome occupancy when assessed on the same sequence.
Yes, they do, but then that becomes a much more extensive question. So when you change RNA modifications, that definitely does change the structure. Unmodified bases make it a more kind of breathable structure. You have a lot more looseness to it. But those changes then also affect other parameters. So unmodified bases make it more prone for double-strand RNA to be present. Double-strand RNA also changes the amount of translation that's occurring, can affect the structure depending on the amount there. So I'd say it's beyond just the presence of the base itself changing the structure. It also changes other components, other impurities that all affect it. And so it then becomes a little bit more necessary to tease apart multiple dimensions to see what's actually leading to any observed change.
And also another question regarding could one characterize both mRNA and guide RNA in one LNP. You know, with the CRISPR and all of that, you have the Cas9 and the guide and so forth. Is this technique, you know, what you guys have in terms of could that be applied to that for understanding all the mechanistic part of what's happening?
Yeah, so I'll answer that on two levels. So for the assays that I showed today, largely that'd be due to it's a fragment base, so the guide RNA is fairly short, and so that's difficult to sequence. Even on a nanopore machine right now, the limit of length makes that hard to look at a guide RNA directly. And so we approach that by looking at the structure of the RNA with and without the guide RNA and looking to see are there changes in base accessibility when the guide RNA is present, indicating the likeliness of an interaction between the two occurring. That being said, there are reports in the literature of different approaches, such as looking directly at RNA to RNA interactions that could give a long enough length for it to come out on sequencing and be able to directly visualize those interactions occurring.
And Mandy, if you have if you have bivalent or a guide strand which is much smaller than the mRNA, could you distinguish between those two in using cryo, or that's something that you guys have not looked into?
Well, I wish. No, but we can see like if there is like how we can recognize in a particle based on a modeled appearance within these particles that there's mRNA present. But we also, for example, do not know what the lowest detection level is. Like how
I
much
see.
does there need to be for us to actually see it? And since there's also no standards out,
Okay.
that if there were standards that with like where you had all particles were full of mRNA, we could use that, but there's not. So it's at the moment very difficult to establish a good tool for this. So we can definitely not see two different mRNAs in a particle.
Okay. So I think we've gone through all the questions and all of that. I'd like to open it for any final thoughts for both of you, you know, in terms of the characterization and where and how this is going to help in the future.
I guess I can get started. I'd say that especially as we're moving towards more therapies that we don't want to be as immune active, like gene therapies, kind of moving beyond the vaccine space, a lot of the approaches shown today from the cryo-EM approaches, sequencing-based approaches, and newer technologies as well are being much more critical to evaluate the full quality before it moves into IND filings and then actually into the clinic. So this is definitely a space that's going to continue to expand as the years goes on.
Yeah, I agree with that. Based on the number of papers that are coming out, like especially this year on the mechanistic function and based on the morphology, etc., I think um, I'm very excited to see the field grow and also to see different methods come out to look at all these quality attributes. And I still think cryo-EM plays an important role in this, but it's exciting to see what's happening in the field. And one thing I would say, and I'm really looking forward to get some standard LNPs out that we can use to develop tools further.
Well, I mean, I do agree. You know, one thing I've said that before and I keep mentioning it. This modality is traveling at the speed of light, and you know, basic technologies that we know of, they're, you know, like having standards, having you know, different things, and understanding certain mechanistic routes and how do you measure it. We're just playing catch up because you know, that modality is just like took off and moving at a very fast pace that regulators, you know, your typical CROs, typical labs, you know, even companies that make analytical instruments and all that, or ancillaries for those are really trying to catch up. But it is very complicated, and part of the reason is the size. You know, we went from small molecule to protein, and everybody wanted to stay away from HPLC at the time because the columns and all of that, the chromatography technology was not supportive of that. Now it have evolved, but if you think about it, mRNA is about 9 times the size of the protein that's translating. Just this is based on codons. You need 3 codons per amino acid. So if you do the math, it's about 9 times the size. So it's way much more larger molecule, you know, in terms of the charge, and it doesn't have this unique structure like antibodies and all of this. So that added more complication. And that's why I'm saying, like, you know, we're moving in the right direction of figure out new techniques to resolve this, because there you can take some from older modalities or previous modalities, but they need to be tweaked a little bit to be applicable here. So you know, with that, I want to thank you both for taking the time this morning. I know both of you on the West Coast, so it's morning for you. So I truly appreciate it. And I do appreciate everyone that dialed in to listen to the presentations and the Q&A and staying with us for the whole thing. So very much appreciated. And hopefully, you know, we'll see you all in about few weeks for the QC release of LNP mRNA drug product. Thank you very much and have a wonderful day or wonderful evening, depending where you are on the in your time zone.
Thank you.
Thank you.